Okay guys, so we're looking today at the scavenger hunt. We want to take a look here. The central dogma scavenger hunt is basically going to take you through a simulation of going from DNA to RNA to protein. When we do that, we'll see that we go from a strand of letters that doesn't make any sense to another strand of letters that doesn't make much more sense to finally a word that we can actually do something with. And the same thing is true with DNA. We've got a string of letters that doesn't mean much to us, but then when it's completely decoded, you get a string of amino acids, which then turn into a protein, and that protein gives you all of your physical characteristics, from your eye color to your hair color to freckles. So if you are interested in doing the central dogma scavenger hunt, you'll notice that you've got 12 problems here. On the next page is what we would call an amino acid chart. There are a few things, though, that we've got to kind of talk through before we can actually dive into this. So when we're looking at DNA, DNA has a few specific things to it. So first of all, within DNA, we've got these 4 letters. We've got A, T, C, and G, and those 4 letters stand for these 4 nitrogen bases: adenine, thymine, cytosine, and guanine, respectively. Now, they pair up very specifically. So A and T will always pair up, and C and G will always pair up. So if we have A, we have it next to a T. We've got a G, we have it next to a C, and so on. A T would be next to an A, and then these things together will form our double helix of our DNA. However, when we switch to RNA, things are a little different. So with RNA, we still have A pairing with T. We still have C pairing with G. We still have G pairing with C. But we don't anymore. I'm sorry. We'll back up here. I gotta back up. Oh, where's my eraser? We have T pairing with A. But when we get down here to A pairing with T, if this is our original DNA strand that we are going to copy, when we copy it into RNA, we'll notice that our RNA no longer has a T, but it has a U. U stands for uracil, and it is found in RNA and not DNA. So this whole chunk then of RNA would break off, head to a ribosome, be read, and become a protein. There's one more step between that. They get read in groups of 3, and that's how we are set up for our central dogma scavenger hunt. We've got DNA in groups of 3. We're going to switch that into RNA, and then switch that into a word that actually makes sense to us. So let me go ahead and make a new page here. Let's take a look at number 1. Number 1 says GCA. So GCA, CTC, CTA, ACT. Now, I'm not going to do the full word because you guys will get the hang of this pretty quickly, but I do want to do enough of it so you kind of see what's going on. So again, this is our DNA strand. We want to convert this into RNA. We have to remember that when we're going from DNA to RNA, A will pair with U, T will pair with A, C will pair with G, G will pair with C. So G pairs with C, C pairs with G, A pairs with U. C to G, T pairs with A, C pairs with G. C pairs with G, T pairs with A, A pairs with U, U, G, A. Now we're going to use our amino acid chart to figure out what letter we're actually coding for. So we now have gone from DNA in blue to RNA in red. Now we need to go from RNA to our protein. So we want to look at our amino acid chart for our little RNA sequence. So the first one we want to look for here is CGU. So I look, CGU is right here. CGU codes for an R. So I would write an R down. My next one is GAG. GAG codes for an E right here. So I would write an E down. GAU, if I look, here's GAU, codes for a D. Write a D down. Red something. UGA is our last one so far. Obviously it won't finish number one, but we'll get awfully close. UGA again, codes for a space. So I'm looking for a red something. Now if we go back here and we take a look at number one, okay, we have these three that we did not decode. Those three come out to be P, E, N. So our protein, or our decoded word for number one, is red pen. As far as points go for this assignment, if you get to the red pen part of this, you earn a 2 out of 3. If you go find a red pen, take a picture of it, and upload the picture, you get a 3 out of 3 for that particular number. There are 12 assignments or 12 numbers here. So this whole thing truthfully, you could, if you wanted to get full credit on it, then you're going to upload me 12 pictures. Okay, and then you'll get full credit based on the picture that you upload. Otherwise, we'll go from there with how many pictures you did not upload. It's completely fine. You will pass the standard if you do just the words. If you get just this far and you don't get the pictures, you do meet the standard. But it's much more fun if you get pictures for it. All right, so that is Central Dogma Scavenger Hunt. This is the last part of Standard 5 here that you guys should be working on. If you choose to not do the scavenger hunt and you do the protein lab, the DNA protein lab is a Glencoe lab. You will need to open that in Internet Explorer in order to get around the Flash issue. You'll be making some amino acids, getting a tRNA code and an mRNA code here for the lab. It's incredibly important that you use the procedure on the left-hand side of the lab when you open it in order to complete the lab itself. Just kind of diving in and not reading that procedure will not make you very successful with this lab. Okay? And then you've got 9 analysis questions, but you do need to have some data here. Okay, so you cannot just do the analysis questions without data. You need to get both. So those are your last 2 options here for Standard 5. Standard 5 is due, I believe, on Wednesday if we take a look at our calendar. I know this is kind of crazy for you guys, but here is Standard 5 for you guys due on the 18th of November. All right, email me if you have questions. Otherwise, have fun with the scavenger hunt. You certainly can work with a partner if you would like. That is totally fine. Just make sure that you both upload your own answers.